Hello, hello. So this is a short video just explaining the stuff for this week. Um, the transcriptional regulation, the Lac Operon, the whole bit. So the hope is to go over all of these things because a lot of people get confused by the videos and by the readings. So the point of this video is to hopefully give you an overall lecture style video of the week 11 material to help you understand it better. Okay. It's out of the way here. Okay, here we go. So this week we're learning about something called the Lac Operon. The Lac Operon is in bacteria. So I guess let me start here. An operon is, the definition of an operon is a collection of genes controlled by one regulatory region. So why do we care? So far in the semester, we've been talking about how you read a gene and make a protein and all of the things that can go wrong with that and how that works. So we've been talking about the what a gene does. But this week, we've started talking about about two other components of the story. We're talking about not just what a gene does, but when, or how do I control when it does its thing and how much protein that gene makes. So in order to do that, we have to have some way to control these genes because the genes don't have controls on them. They're just the instructions on how to build proteins. And the enzyme RNA polymerase, we learned about in week 5, RNA polymerase, it comes in and it reads the DNA and it transcribes it into RNA, which then gets translated into protein. But so we need an RNA polymerase, enzymes kind of have blinders on. So they do one thing and that's their job in life. And they don't really care about other people's opinions. So it doesn't matter what the need is. RNA polymerase, just like every other protein and enzyme, just does its thing regardless of circumstances around it. So RNA polymerase, all it knows how to do is it sticks onto this area of the DNA called the promoter. And I remember that because a promoter is like somebody that, a promoter for an event is like somebody that promotes the event, tries to get people to come to it. So the promoter is the one that brings RNA polymerase to the party. So the promoter is just a DNA sequence and the RNA polymerase sits on top of it. So it clicks onto the promoter and then RNA polymerase, just like a train, choo-choos down the train track, choo-choos down the DNA train track till it gets to these genes, and then it reads the genes, transcribes them, and then they eventually get translated and turned into proteins. So these proteins that we build, so let me go here. So the RNA polymerase goes down and it builds, and we'll just sum it up in saying that these 3 proteins result. These 3 proteins, beta-galactosidase, lactose permease, and transacetylase, have a specific function in the cell. In particular, these ones are in charge of digesting a sugar called lactose. And most of you are familiar with lactose in relation to milk. Lactose is a sugar that is most commonly found in milk and other dairy products. And it's also well known for lactose intolerance. And I'll explain a little bit of background behind lactose intolerance later on in the video. So here's the story behind this. Turns out bacteria and humans, so basically all cells, are designed to eat a sugar called glucose. Glucose is a very simple sugar, and all life is designed to eat that. That's our main food source. And in fact, it's so strictly defined that we can't use anything except for glucose for energy. So we have to be able to, so anything else that we eat, the cell has to be able to convert it. If it's going to use it for energy, it has to convert it over to glucose somehow to be able to use it. Even other sugars have to be converted into glucose, otherwise they won't work for energy. So the point of this operon is to make these proteins, and their job is to take on lactose. So this looks really complicated. This is the molecular structure of lactose, but I've simplified it down to here. Basically, it's two hexagons holding hands. That's basically what it is. You don't need to know this. This is just explaining the background here. So the basic idea is it's two hexagons holding hands. Each hexagon is essentially a glucose molecule. One of them's called galactose, but it can be easily changed into glucose. So essentially lactose is two glucose molecules glued together. That's what lactose is. Each hexagon is glucose. But just like how you can't eat the entire ham or something like that, we need to cut it down into the little pieces, because even though lactose is made up of glucose, because they're glued together, the cell can't do anything about it. It can't use that for energy. It can't burn it for food. So in order to use lactose as an energy source, the cell has to come in here and cut the two of them apart. Now I've got two glucose molecules and I can burn those. So the whole point of these three proteins over here, oops, the whole point of these three proteins over here is to cut lactose in half to make glucose so that the cell can burn it for energy. That's the whole point. So this entire setup is about making proteins that are capable of turning lactose into glucose. The problem is RNA polymerase, like we said, it puts its blinders on and it just does this all day, every day. So it hops on at the promoter and it goes, reads the genes, and we end up with the proteins. Then it hops off, comes back down here, lands on the promoter, and does it again and again and again, all day, every day. That's all it knows how to do, which means we'll be constantly making these proteins. Now that's bad. Not with all proteins, but in this case, that's bad because it takes a lot of energy to build a protein. So we don't want to waste energy building proteins that aren't going to help us. If you think about it, these proteins, their job is to destroy lactose and turn it into glucose. So if there's no lactose for them to destroy, then building the proteins is a complete waste of energy, because they're just going to be built and float around and do absolutely nothing. And I just wasted all the energy I used to build those proteins. So I want to be able to control when these proteins are made so that RNA polymerase only makes them when lactose is there for them to chew on, and stops making them when lactose disappears. But remember, RNA polymerase doesn't take orders from anybody. He just does his blinders thing. He hops on the promoter and goes all the way to the end. And then he hops back on the promoter and goes all the way to the end. So somehow we have to find a way to stop him. So we created another protein. Well, God created another protein, but we'll add another protein to this system called the repressor. And I don't know if you've seen Monty Python. If you haven't, this isn't gonna make any sense. But on Monty Python, if you've ever seen that one, the king is trying to stop the peasant from his giving his speech, and so the king goes and tackles him, and then the peasant's like, help, help, I'm being repressed. When you repress something, it's trying to shut it down, prevent it from happening. So the repressor is a protein that's designed to shut down the Lac Operon. The way it does that is, again, proteins have their one job in life. The repressor's one job in life is to grab onto a DNA sequence called the operator and hold on tight and not leave. That's its job in life. Its job is to hold on to that and just stay there. And this repressor is this big, bulky protein that gets in the way. Basically, it's a linebacker. The idea of a linebacker is you put this big guy that's got a lot of muscle and he's heavy, so that the other guy runs into him and nothing happens. He just stays put. So the repressor, we're essentially, going back to my train track analogy, we're essentially putting this great big block on the train tracks. And so then now when RNA polymerase hops on at the promoter, he starts chewing down the train tracks and runs smack into this brick wall. And doesn't make it to the genes. He can't make it to the genes. He'll hop off, hop back on the promoter and try again, run back into the wall. As long as that repressor is there, as long as the big brick is in the way, RNA polymerase can't get to the genes. We just turned off the operon. Essentially, we just cut off access to the operon. So this is the off switch, basically. Think of it, you might think of it like a light bulb. If these are the light bulbs and RNA polymerase is the electricity, if RNA polymerase is always getting to the light bulbs, then we're going to turn on the light bulbs indefinitely. Now I just cut off the electricity, so I added in a switch that's able to shut off the electricity from accessing the light bulbs, and now the light bulb's not turning on. This state where we're not making proteins is called, we say that the Lac Operon is being repressed. So when proteins are being made, the operon is activated. When proteins are not being made, the operon is repressed. Proteins equal activated, no proteins equal repressed. Okay, so in this case, it's repressed because RNA polymerase can't get from the promoter to the genes because there's this big brick of the repressor protein in the way. So that's great and all. I figured out how to turn it off. But now what? We have to be able to turn it back on again. The point of a light switch is not just to permanently turn it off. We need to turn it back on again, but the repressor is holding on to the operator. So as long as he's sitting there, we can't turn this thing back on. So we've got to somehow have a way to get the repressor out of the way when we need to make the proteins and then put him back again when we don't want to make the proteins. So God is a genius. This thing is basically a padlock. Think of the repressor, we've padlocked it to the operator and it's not leaving. But how do you get rid of a padlock? You have a key. And by inserting a key, it causes the lock to change shape, which unlocks it and makes it let go of whatever it's locking. Same way here, we need a key to get the repressor to leave the train tracks. And the key, this is why I say God's a genius, the key is extremely intuitive. If you think about it, why did we want these proteins in the first place? The point of the proteins was to come down here and chop lactose apart. So we need the proteins when lactose is there. So God was a genius because he made lactose the key. That way we only unlock it when lactose is there and we relock it again when lactose disappears. Cool, huh? So when lactose comes in, lactose sticks to the repressor and it makes the repressor let go of the operator and it floats away. Now there's no longer a brick on the operator. So RNA polymerase hops on at the promoter and keeps chewing down the track and starts building the proteins again. He comes back over, and as long as there's lactose, that repressor will stay up in the air and RNA polymerase will just keep making proteins until all the lactose is gone. Now, here's the fun thing about this. It's a self-regulating system. God designed it to be a switch that turns itself back off again. So these proteins, remember we determined that the point of these proteins was to destroy the lactose and turn it into glucose. So lactose is the key. We're destroying all the keys. So essentially, this system is designed to turn on so that it has the ability to turn itself off again, because these proteins destroy the key. And if you destroy the key, the lock goes back onto the operator and shuts it back off again. So see, it's a self-regulating system, kind of like those new motion sensor light switches. It turns on the light when you're in the room and it turns off the light when you leave. That's how it works. It's on when the lactose is there, and once all the lactose gets destroyed, then it goes back off again because the repressor gets back in the way of RNA polymerase, and now it's off until we need it again. So that's the idea of this switch that allows us to turn on, activate gene expression, and turn off gene expression. Gene expression meaning we're making proteins off of these genes. So by adding lactose and removing the repressor, by adding lactose, which allows the repressor to leave, we activate the genes. And by removing lactose, we put the repressor back, which represses the genes. In this role, lactose is what we call an inducer. There are lots of genes in the body, and not all of them are influenced by lactose. But all of them, there's some molecule that acts as the key. And the key is what we call an inducer. The key, whatever the key is, lactose in this case happens to be the inducer. It's whatever the inducer induces the expression in the operon. It's the thing that causes it to happen. So you need the inducer which is lactose in this case, to bind to the repressor to get it off the train tracks so that we can activate expression. So that's everything you need to know for your homework, because all the homework is, problem 1 is just eliminating certain ones or certain of these 3 aspects and being like, well, is it going to be activated if only these 2 pieces are here and not that one, or will it be repressed? What about these ones? And the idea behind that is showing the influence of each of the pieces. And a rule of thumb for you is, remember, how did we turn this thing off? Well, if the repressor's in the way, then it's repressed. And if the repressor's not in the way, then it's activated. So if for any reason the specific combo you get on your homework results in there not being a repressor in the way, whether it's lactose peeling it off or the repressor physically not being present, it's activated. And if there is a repressor in the way for any reason, or for any reason RNA polymerase can't make it to the genes, if for any reason RNA polymerase can't make it to the genes, then it's repressed. Because remember, activated, proteins made. Repressed, proteins not made. Okay. All problems, the only difference on problems 2 and 3 of the homework is a lot of students get confused on this one because we say that it's the Lac Operon, but then we've got this gene, this GFP. We're saying that we're making GFP, and it's like, wait a minute, GFP is not one of these genes. Well, it's true. GFP is not part of the Lac Operon, naturally. But as scientists, when we're trying to figure out how the operon works, let's pretend it's back in time and we don't know how this thing works. We don't know what turns it on. We don't know what turns it off. We just know that there's a DNA sequence here and something's got to influence it. We have to be able to see the results. If you flip on a switch and you see the light bulb turn on, you know the switch turned on that light bulb. If you flip off the switch and the light turns off, you know that switch controls that light bulb. But the problem is these 3 proteins are invisible. So we actually can't tell the difference visually between a bacteria that has an activated Lac Operon and a bacteria that's a repressed Lac Operon. So I can't tell what turns on the operon and what doesn't. So what we do is we use the methods we learned last week with gene cloning, and we take the GFP gene, and basically what we did was we cut out these 3 genes and replaced them with the GFP gene. Essentially what we're doing here is we're tricking the light bulb, or we're tricking the system. The cell still thinks that flipping this switch is going to make the proteins that will destroy lactose. So when lactose comes in, it still flips the switch just like normal. I just changed what flipping that switch does. I changed the light bulb that is activated. So I swapped out a white light bulb for a red one. So I flip the switch, it's no longer white, it's a red one. Ta-da! So instead, it makes the GFP gene. It makes the GFP protein. Now, if you remember from week 10, GFP stands for green fluorescent protein. It's a protein that glows in the dark. Green, conveniently. Now, that we can see. So this serves the role of a reporter gene. We actually don't cover reporter genes, so that's not one you need to know. But the idea behind this, it's kind of similar to the selectable marker, where the selectable marker helped you see the difference between a bacteria that had the plasmid and a bacteria that didn't. The reporter gene is helping you tell the difference between a bacteria that has turned on the gene and a bacteria that has turned it off. So if the bacteria turns it on, or turns on the operon, it's going to make green fluorescent protein and glow. So if I add lactose and the bacteria starts glowing, I'm like, aha, lactose caused it to turn on. And if I take away the lactose and it stops glowing, then I'm like, aha, removing lactose turns it off. Now I know how it works. So, but problems 2 and 3, it functions the same way. If you can make it through problem 1, you know how to do problems 2 and 3. We're just changing what the light switch turned on. So don't get confused on that one. GFP is not part of the Lac Operon naturally. We added it in there so that we could see when the light switch was turned on and when it was off. Okay, so there's the basic idea there. Let me switch back to the natural Lac Operon. Now, that's everything you need to know for your homework, but there's more that you need to know for the week. So we're gonna add in a few other components, because if you remember, there were two questions that we were trying to resolve this week. One of them was, how do we control when the gene is transcribed, which we just solved, the repressor. The repressor allows it to be made or not, and we're able to control whether it's made or not. But the other question was, we want to control how much of it we make. If you think about it like in cooking, we want to be able to control when we start making dinner and how much of dinner I make. If I'm trying to feed 8 people, I don't want to make just 1 plate's worth of food. And if I'm feeding 1 person, I don't want to make an entire buffet to feed the county. I want to be able to control how much of the protein products I make in order to, or depending on the circumstances. Because remember, I want to use as little energy as possible. So I don't want to make any more of these proteins than I need. And the repressor, it doesn't have any influence on how much protein we make. It just has influence on when we make it. So we need to add in another piece to this puzzle to help control how much of the protein we make when we're making it. So we add in a few new components here. In addition to having the RNA polymerase and the repressor, we're going to add in another protein here called the activator. The activator, so for the Lac Operon, the activator's name is CAP, stands for catabolite activator protein, but you don't need to know that. I'd focus more on the name activator. The activator, it's the protein responsible for controlling how much we make, how much of these proteins we make. I like to think of the activator like a cheerleader. If you think about it with the marathon runners, you know, they run at a standard pace without anybody cheering them along. They're just running at their standard pace. But then near the end, there's that guy that's always standing on the corner that's like, go, you're almost there! Like, run a little faster! And so then the marathon runner's like, yeah, and he speeds up. Well, the goal here is, in order to make more of these, I need to speed up RNA polymerase so that it goes through its cycle faster. And the way, so it goes through its cycle faster and more often, hence it makes more protein in the same amount of time. And the activator, when it's bound to its own little sequence, so notice the repressor binds to the operator. And I like to remember that because like telephone operators, they're the ones that can end a call. So repressors, the repressor binds to the DNA sequence called the operator. RNA polymerase binds to a DNA sequence called the promoter, and the activator binds to a sequence called the enhancer. And the enhancer and the promoter and the operator together form what we call the regulatory region. So over here, the 3 genes, these are the genetic region. And these 3 components over here, the enhancer, promoter, and operator, these are the regulatory region. And the regulatory region plus the genetic region is the Lac Operon in total. So regulatory region plus genetic region, switch plus light bulb, that's the Lac Operon. Okay, sorry for the detour. So back to the activator. When it binds to the enhancer, then it speeds RNA polymerase up and will make lots of the protein. But unlike the repressor, so the activator is kind of backwards of the repressor. The repressor, its whole job was to hold on tight to the DNA until the key forces it to let go. The activator is the opposite. It's a slacker. And so normally it just floats around in the water, not even touching the operon, not doing anything until its key comes and drives it on. So it's like a lazy bum that you have to force to go do his chores. The activator just floats up here not doing anything until it gets its key, which then forces it to bind to the enhancer, and that makes things speed up. So the key for the activator is another inducer, because remember all the keys are inducers. So the key for the activator is a little molecule called CAMP. It stands for cyclic adenosine monophosphate, which again, you don't need to know. And the weird spelling, or the weird capitalization here is on purpose. It's legit little c capital AMP. The funny thing is, this is actually just a slightly modified adenine nucleotide from DNA, but the cell uses these as signal molecules. When CAMP binds to the activator, then the activator binds to the enhancer, and then RNA polymerase speeds up. So if I get the repressor out of the situation here, when I do that, RNA polymerase is going to make lots of protein because the inducer made the activator stick to the enhancer. If we don't have the inducer, we don't have CAMP, then RNA activator doesn't stick to the enhancer, and RNA polymerase just makes its normal amount. But with CAMP, then we're going to make twice the amount. Now, here's the logic behind this, because there's reasoning for this, and this will explain where CAMP comes from. CAMP shows up when glucose is missing. Remember how I talked about at the beginning of the video that glucose is the kind of food that, or glucose is the food that the cell knows how to eat. So if it's sitting on top of glucose, then it's like, heck, why do I need to go through this extra step of preparation? Why do I need to transform something into glucose if I already have lots of glucose just naturally here? It's like, why would you cook dinner if you could eat out for free all the time? Like, it just seems like a waste of energy. And so we're not going to want to use lactose. We're not going to need to eat that much lactose if there's tons of glucose to fulfill the need. So we'll want to maybe snack on the lactose to help supplement the amount of glucose that I've got. So we're going to make a little bit of the protein, but we're not going to want a ton. The problem is when glucose goes missing. If glucose stops being in the environment, so the bacteria has eaten all the glucose around it, and it's like, shoot, its options now are starve to death or turn something into glucose. And so it panics and sends out its panic button. And CAMP is the panic button. The idea behind CAMP, so the Lac Operon is one of several operons, and each one takes on a different sugar. The idea of CAMP is it's like we've got all these backup plans. We can digest lactose to get glucose. We could digest a different sugar to get glucose. And so we send out CAMP to go check all of the backup plans to see which one of them we can start using to turn into glucose, to turn that sugar into glucose so we can use it for energy. So when CAMP comes spitting out, if glucose is gone, the reason why we want more of these is we can no longer afford to have lactose be a side dish. It has to be the main course, otherwise we're going to starve to death. On a bacterial level, this bacteria is going to starve to death without having glucose. So we make lots of these proteins because now the lactose is the main course. And we're going to keep doing that until glucose comes back in sufficient quantities that we stop sending out the panic button. And then the CAMP will go away and we'll start making the nibbling amounts again. So that's how the activator in CAMP works. So don't get confused. CAMP has an inverse relationship with glucose. The less glucose you have, the more CAMP. Does that make sense? The less glucose you have, the more CAMP you have. And the less CAMP, the more glucose. So that's how that works. Now, the Lac Operon, it participates in a system called combinatorial regulation. What this vocabulary term means is there are multiple proteins that have a say in regulating when these genes are transcribed and when they aren't. We've talked about them. We talked about how both the repressor and the activator, based on circumstances, have a say in when these proteins are made and how much of them. So the Lac Operon is an example of combinatorial regulation because there's a combination of factors that control the making of these proteins. So that's a good vocab word to know, combinatorial regulation. So it's interesting doing like a problem 1 on your homework style question here, where we're like, what if we have the activator and the repressor and RNA polymerase and lactose and no glucose? So that means CAMP is here. And that means lactose sticks to the repressor, gets the repressor out of the way, CAMP sticks to the activator, puts the activator on, and we're going to make lots of protein. So notice it's a combination of factors. What if we have lots of glucose and lots of lactose? Then we would have we would have everything except camp because we've got lots of glucose, so we're not sending out the panic button. So lactose will get the repressor out of the way, the activator will stay off the enhancer, and we'll just make a little bit of the protein. Now, here's the interesting thing about this. What if we have no glucose and no lactose? So we've got our 3 proteins here. The no glucose says camp is here. Oops, sorry, I pressed the wrong button. The no glucose means camp is here. And the no lactose means the repressor is still stuck to the operator. So what's going to happen? It's repressed. Because even though this one's screaming, go, 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 we got to digest something to get food. This one over here is saying, but there's no lactose to digest. Remember, the point is to save energy. And so even if the cell's starving to death, if there's no food in the lactose sector, then we're going to be wasting energy building these things. So those other operons working on a different sugar, one of them is going to have to take up the camp message and go build its protein to take down whatever sugar is available. But the Lac operon in this particular case won't be helpful to the cell because even though there's no glucose, it can't help the cell because there's no lactose either. And that's its only job, is taking down lactose. So the repressor is a permanent shutdown switch, or is a shutdown switch, and the activator can scream as much as it wants, but the RNA polymerase can't get by until the repressor gets out of the way. So it's a combination of factors that allows us to control when we make the protein and how much based on our circumstances. So that's how the Lac operon works. And I encourage you to go, and my simulator's on my website. I encourage you to go play with this. Go practice getting all of these down. Remember that that the enhancer, the promoter, and the operator are part of the regulatory region. Lac Z, Lac Y, Lac A are part of the genetic region, and those all make up the Lac operon. Now notice over here on the left, there's another component I haven't talked about. That is Lac I. Now Lac I is a gene, but notice it's not part of the Lac operon. It's elsewhere. I don't know fully where, but it's elsewhere on the bacterial's DNA, on the bacteria's DNA. Lac I is the name of the gene that makes the repressor. So the gene, so Lac I has the instructions that when it's transcribed and translated, it makes this repressor protein over here. But a lot of people get confused and think that Lac I is part of the regulatory region. It's not. It makes a regulatory protein, hence we call it a regulatory gene. Lac I is a regulatory gene, but it is not part of the regulatory region. Let me say that again. Lac I is a regulatory gene, but it is not part of the regulatory region or by extension the Lac operon. It contributes the repressor protein. Without Lac I, you can't have the repressor, but it's not part of the operon. Okay, so there's that one over there. One other thing one other regulation method that we use is called DNA methylation. Now this one, you would rarely see it with the Lac operon itself, but knowing that it exists and what it does is important for this week. DNA methylation is a process where an enzyme that called DNA methyltransferase, but you don't need to know the name of that, comes in under certain circumstances and glues these little, glues basically a carbon atom to all of these genes at a bunch of different spots. It's called methylation, not to be confused with methamphetamine or meth, the drug. It's called methyl. So we're methylating the genes, and what that does is it basically sticks these little methyl groups to the genes. And when you stick the methyl groups to the genes, it permanently shuts down the genes. It's repressed forever. Now with the case of the Lac operon, that's not a very helpful thing. So we wouldn't see that on the Lac operon, but on other genes it can be. It can also cause problems. So yeah, in this case, even if let's say for example, we did methylate the Lac genes over here, even if we had no glucose and mountain loads of lactose, it's not gonna transcribe the genes because we methylated them. We shut them down. It's like sealing 2/3 of the Book of Mormon. Doesn't matter how badly we need to read those 2/3, until God says we can open up those 2/3, we can't open up those 2/3. It's sealed. So the instructions are there, we just can't get to them. And so it's permanently repressed. By methylating a gene, you permanently repress the gene. And that's for whatever gene. If you methylate the Lac I gene, that means it will be permanently repressed, so it won't make the repressor. So that means this will be permanently turned on. So for example, if we take away that one, but we methylate this one, and remember the Lac I, it was supposed to make the repressor, and if it's methylated, it's not going to make the repressor. So that means there's nothing to ever turn this gene off. And so we'll constantly make it. We have another vocab word for that. The vocab word is constitutive, spelled constitutive, and there's probably 1,000 ways you can pronounce this depending on what part of the country or the world by extension you're from. But it's constitutive expression. What that means is these genes aren't regulated anymore. It's constantly activated. In this case, there's no repressor anymore because we methylated the Lac I, and so the Lac Operon would now be constitutively expressed because there's nothing to stop RNA polymerase from doing its job all the time, day and night, 24/7. With the Lac Operon, that's bad because we want to be able to turn this thing off. With other genes, that's okay. For example, RNA polymerase. RNA polymerase is a protein, which means there's a gene for RNA polymerase. So RNA polymerase builds other RNA polymerases. That's kind of interesting to think about. But we need RNA polymerase all the time. The cell is constantly making proteins, and so it constantly needs RNA polymerase. And so we don't ever need to turn off the RNA polymerase gene. So the RNA polymerase gene doesn't have a repressor. There's no repressor. It just has a promoter and the gene for RNA polymerase. And so it's constitutively expressed, and that's a good thing, so that we always have enough RNA polymerase to build all the proteins we need to keep the cell alive. But in other genes, it's bad. If we have constitutive expression, for example, think of it like this. Let's pretend these genes these genes over here were genes that control when the cell divides. So if these proteins come out, they tell the cell to divide, go through mitosis, and make a baby cell. We would want a repressor. So let me take away this methyl over here. We would want the repressor to come in here and to, we want the repressor to make sure that these proteins are made when the cell's ready to divide, and that they're not made when the cell isn't. But what if we get a mutation in the gene that makes the repressor for those for those dividing genes? Then that would mean either that or we methylate that gene. But more commonly, it would be a mutation. If we ruin that gene over here, it won't make the repressor anymore. That means there will be nothing to stop these guys or the proteins that would be dividing the cell from being made, and so the cell would constantly divide, and we call that cancer. That's one of the ways you can get cancer, is by causing constitutive expression of proteins that are responsible for dividing the cell. And so when they're constitutively expressed, there's no way to stop the cell from dividing, and so we call it cancer. Let's see. So the last thing for me to cover here is, let's see. Actually, let me do the story here with the with lactose intolerance. I promised you I would go into that one. Humans, we don't have operons. Bacteria are kind of smart. They put multiple light bulbs on one light switch. They say, well, if I need all three of these proteins to digest lactose, then why not just put them all on one switch? Cells like ours, eukaryotes, we're less efficient. We like our control. And so we have a light switch for every light bulb. Every single gene has its own regulatory region. And so, but for humans, we've got something very similar here. Rather than using these three proteins, we have an enzyme called lactase that does the same exact job. It converts lactose into glucose. And so our lactase gene over here is making a protein to go and digest lactose when you drink milk, and then it stops making it behaves the same way as the Lac Operon. And if you think about it, lactose intolerance means that they're not able to tolerate lactose. Their body does weird things when they drink milk. It causes them stomach pains and diarrhea and bloating and all sorts of things. Well, if you think about it, almost all lactose intolerant people drink mother's milk as a baby. They weren't allergic to lactose then. There's milk in mother's milk, or there's lactose in mother's milk. So what happened? They obviously have a working lactase gene. Otherwise, they would never be able to make that, and they would have lactose intolerance from the very beginning. So they have a working gene. It's not a mutation that's the problem, but then they gradually, when they get older, at some point, they become lactose intolerant. Now, there are a couple of ways you can do that. It could be an allergic response, or things like that. But one of the ways you could do it is, one of the most common ways is DNA methylation. Turns out humans are not supposed to be lactose tolerant. We're supposed to be lactose intolerant, because what idiot decided that it was a good idea to go drink another animal's mother's milk? That's just a ridiculous notion, and yet we do it all the time here in America. But that's not the case all over the world. A lot of places culturally don't drink as much milk as we do. So a kid gets weaned off from his mother, and he just doesn't need it, or he doesn't have milk anymore. And so then the body is like, well, we haven't used this gene for 3 years. Let's methylate it. Let's shut it down, get it out of the way. So we methylate it, we shut it down, we get it out of the way. Gene is methylated, we can never use it again. Great, that's fine, we don't plan on using it. Then the kid moves to America from China, and he drinks his first glass of milk. Now the lactose comes in, and the cell's like, freak. We need something to digest this lactose stuff, and it goes through its records, and the gene's methylated. It's permanently sealed. We can't get to it anymore, so we can't make that protein anymore. So now all of a sudden, your body doesn't know how to digest lactose. So then it stays in your intestinal tract. The lactose goes down to the large intestines, and the bacteria that live in your large intestines have the Lac Operon. So they start eating the lactose, have a heyday down there, and in the process of doing that, they give off gases and other things that make you have diarrhea and bloating and pain, hence lactose intolerance. But the idea behind it is you can methylate a gene, permanently shut it down, and that's one way you can become lactose intolerant. But the cell also uses DNA methylation for good things too. For example, if you think about it, all cells in your body came from the same cell. A sperm fertilized an egg, and that cell had 46 chromosomes, and it divided and gave 46 chromosomes to its kid. So every cell in your body has the same genetic information. They all have the same book. So why does your skin not ooze bile? Because your liver does. The liver cells make bile. So why doesn't your skin? It's got the same instruction manual. And why doesn't your nerve excrete bile? Why are your bone cells different? They all have the same instruction manual, but what happens is when that cell's dividing, we start getting to a point in the development of the baby that where the cells make career decisions. It says like, okay, I'm going to be a liver. And so it goes through the whole genome, all 23,000 genes, and finds all the ones it doesn't need to be a liver, and methylates them all, shuts them all down permanently, keeps the ones it needs to be a liver, shuts down all the rest. So essentially it only has access to the recipes it needs to be a liver cell. The skin cell does the same thing except for skin cell genes. So it shuts down everything it doesn't need to be a skin cell. It methylates them all. And so as they make these career decisions, they become more and more specialized by shutting down more genes with DNA methylation. And ta-da, you wind up with cells that have the same genetic information but behave differently because they have access to different genes. You don't need to know that for this class, but I think it's a lot of fun to think about. Okay, last point here, and then I'll let you go. There's another vocabulary word that people overlook is a consensus sequence. A consensus sequence is any sequence that is found in more than one or in many regulatory regions. If you think about it, because like the word consensus, if a group comes to a consensus, they come to an agreement, they all agree, everyone has the same idea. So a consensus sequence is a sequence that is found in the regulatory region of basically every gene. An example that we ask you to know about is the sequence TATAAT. I know it sounds very specific. We call it the Tata box. That's its nickname for obvious reasons. TATAAT is an example of a consensus sequence, and it's found in every promoter of the cell. So the promoter is a DNA sequence. TATAAT is found in every promoter. The reason for that is TATAAT is the sequence that RNA polymerase binds to in the promoter. And we use the same RNA polymerase to transcribe every single gene. So the RNA polymerase has to recognize the promoter, or has to recognize every single promoter for every single gene in the entire cell. Hence, its recognition sequence has to be in every single promoter. That's why it's a consensus sequence. Every promoter for every gene has it. So TATAAT, the Tata box, is the consensus sequence we ask you guys to know. But the idea behind a consensus sequence is that it's a sequence found in the regulatory region of more than one gene. So yeah, that's a sizable chunk of this week. Sorry to dump it all into one big long video, but hopefully that helps. Please reach out to me or your TA or teacher if you need more understanding, and please take advantage of the Lac Operon simulator. It's a lot of fun.